So I'm here at Newground Coffee in Headington, enjoying a wonderful iced latte with Dr Matthew Baxter, who is a researcher and fellow of the British Heart Foundation. So your lab studies coronary heart disease and the genes which affect it, but how do you know which genes actually do affect it?
So we do something called genome-wide association studies, or GWAS, and that's just a fancy term for sequencing DNA from thousands of people and then finding which sequences correlate with how you get the disease. Most of the variations which correlate with heart disease fall into the non-coding genome.
And is that what people used to call junk DNA?
Yes, it is what people used to call junk DNA. So the non-coding genome basically means the bits of our DNA which do not code for genes which are translated into proteins. So this non-coding DNA makes up about 98% of each of our genomes. And for a long time, it wasn't clear what these bits of DNA did or if they had any function at all. But increasingly, it's becoming clear that they're extremely important for turning genes on and off. So a better word than non-coding DNA might be regulatory DNA, because they regulate where and when our genes get turned on and off.
That's quite an abstract concept. How does this actually happen in practice?
So these regulatory bits of DNA can be located hundreds of thousands, even a million base pairs away from the genes which they target, which in terms of DNA is miles away. And what actually happens is that they form these three-dimensional loops in space to bring these regulatory elements of DNA close to their target genes. And through these physical interactions, they can turn them on or off.
I didn't realize DNA was quite so dynamic.
It's very dynamic.
So we know that your genes determine a lot of your characteristics, right? Or they at least affect them. So if you took my DNA, my genome, would you be able to tell what I look like?
No, no chance at all. I could build a statistical model to make a prediction about maybe a handful of your features, but the model would be very inaccurate. So no, really no chance at all. And that's because of the interaction between the environment and your genes, which is why my lab focuses on these non-coding regulatory regions of DNA. And we're really interested in how these elements interact with the environment.
So essentially, biology is complicated.
Very, very complicated.
Okay, so the non-coding genome is important for regulating the coding genome, so what actually produces proteins.
That's exactly correct, yes. And a lot of the variants or the vast majority of the variants which correlate with heart disease fall into this non-coding regulatory DNA elements.
Right, so if you have the DNA variant in your genome, does that mean that you will definitely get the heart disease?
No, virtually none of the DNA variants that we commonly inherit will guarantee that you get heart disease. Most of them increase or decrease your percentage chance of getting the disease only by 1 or 2%. And your genes also interact a lot with the environment. So it's combinations of your genes and environmental risk factors which ultimately determine the percentage risk you have for a disease like coronary heart disease.
Okay, so it's like we talk about the nature-nurture debate. So it's the genes and the environment that contribute to heart disease. So what type of environmental factors can increase your risk of heart disease?
Probably the main two are cigarette smoking and exposure to high cholesterol. High cholesterol has been shown to reprogram how your immune cells work. Exposure to cholesterol activates some of these non-coding regulatory elements we were talking about, and it leaves a chemical signature on them. And this chemical signature can persist for many years after your cholesterol levels are lowered again. So these regulatory elements of the DNA which are labeled or modified by the cholesterol exposure can then change the way your cells behave by changing gene expression for many years after you lower your cholesterol.
Are you telling me that if I eat McDonald's, it can affect my genome?
I think one McDonald's will have a very limited effect, but over a long period of time, yes, everything that you eat, everything that you drink, everything that you do will ultimately affect these regulatory sequences in your DNA.
So it's really interesting that science now seems to be advancing into more of a holistic idea of what affects your characteristics. So it's not just genes, it's not just environment, but it's a combination of both. I think that's fascinating.
Yes, it really is. It's a combination of both, and the number of combinations is almost infinite. So it's impossible to predict the effects, even with the best supercomputers that we have.
Okay, so I'm really interested in knowing how you actually ended up in academia. Was it always your plan? Did you always want to be a scientist? How did you actually end up here in Oxford?
So the honest truth is that I started a PhD because I didn't want to get a real job. And then after I finished my PhD, I worked in sales for a year or so, and I realized that really didn't suit me. And what I really craved was the academic environment. So I managed to work my way back into science through a postdoctoral position at the University of Manchester. And then a few years later, I moved to the University of Oxford, and I've been doing genetic research ever since.
That's great. It's such a non-traditional path, but I think that that's something that should be talked about a little bit more, because sometimes it's not quite as linear as school, university, science.
I don't think there's such thing as a traditional path. Everyone I know and work with and speak to has what people would call a non-traditional path.
That's super interesting. I guess academia is quite a non-traditional profession.
I suppose it is, yeah.
So, what was your most exciting discovery yet?
I think the most exciting discovery yet is using machine learning and some advanced molecular biology techniques. We discovered how these genetic changes in your regulatory DNA affect the cells which are inside your blood vessels in order to cause them to dysfunction and accelerate the progression of coronary heart disease.
Okay, so it's been really great chatting to you. I just wanted to ask you one last question. What advice would you give to any junior or aspiring scientists who might be watching?
I think the most important thing is to get a really great mentor. And I think this is important at every stage of your career, but especially when you're a junior scientist. Having a really good mentor can really accelerate your progression, and having someone in your corner looking out for you really helps.
That's great. Thank you so much for meeting with me today. It was really interesting.
Thank you, Grace.